thanks to the other speakers who've given some fantastic talks and great introduction to set the scene for this talk on the pipeline that we've written in Public Health Wales to analyse SARS-CoV-2 sequencing data. My contact details are down here. Feel free to get in touch if there's anything that I say today that isn't clear or you'd like more information on. You can @ me, you can email me, you can contact me on the Slack. I guess I'm going to start with some acknowledgements here. This is quite a difficult one to do for me because I'm not really used to giving pipeline talks. This is all new stuff. So really, I wanted to acknowledge all of the bioinformatics open source software, because without them and their efforts, none of this this pipeline or any of the amazing work that COG UK has been able to do over the last year would have been possible. So the pipeline that I'm going to talk about now depends very heavily on the ARTIC pipeline that Will's described, and Will and Nick were instrumental in building that. So this covers the Nanopore side of the pipeline, and the Illumina side of the pipeline that I'm going to talk about is mostly IVAR is fundamental to this to the Illumina side of this, and this is a tool written by Christian Andersen's lab, particularly ARTIC, but also Allah recently has been working very hard on improving IVAR for use with SARS-CoV-2 and the ARTIC protocol ARTIC lab protocol that Josh described. And then anybody that's contributed to the Nextflow repo or the GitHub repo that the Nextflow code for this pipeline is on. So this is particularly Keith Jackson and his team at Sanger. Anybody that's opened an issue, anybody that's even better made a pull request, anybody, and I think lots of people have forked this initial repository and taken it and made it their own and added things that they needed for their sequencing pipelines to it. And also not to forget the people that are packaging the software for release. This is a pretty thankless job, and the time and effort that goes into doing this is huge. And yeah, a big thanks to anybody that's ever packaged a piece of software for Conda or Bioconda. This is my level of graphic design, so it's a bit of a step down from Will, but I wanted to put this in to sort of illustrate where this talk is pitched in the process of traveling from a sample, so RNA for SARS-CoV-2 to a consensus FASTA, which is kind of the currency of sort of exchange for bioinformatics in SARS-CoV-2. You can take this and you can upload it to GISAID, you can type it, you can do phylogenomics on it, all kinds of stuff. And the sample travels from RNA, it travels through Josh's talk, where we talk about the amplification of reverse transcription and amplification of the sample. Then sequencing happens in Public Health Wales. It's a very dedicated team of people that turn samples into fast queues, and then some sort of wibbly line stuff happens. Some bioinformatics happen, and at the other end, you get your consensus sequence FASTA. And I'm going to talk today about the sort of travel, the process of traveling from a FASTQ file that comes out of a sequencer to a consensus sequence FASTA. So, why build a pipeline? Is the first thing that I want to justify my existence by describing why we should build pipelines. And I'm going to use this pipeline as an example of that. And then I want to talk a little bit about workflow managers and workflow management languages, and why you should use one. And probably this part of the talk is going to be 80% me evangelizing about how great workflow managers are and how important they will be in the future. If you're just getting into bioinformatics now, please learn a workflow management language, because they're going to be, well, they already are really key to reproducible research, and they will only become more so in the future, I think. Then I'll talk a little bit about what the pipeline does. There'll be some horrible lines and boxes of pipeline diagrams in there, so my apologies for that. And then finally, I'll talk very briefly about how to run the pipeline on your data, because most of this information is available in the GitHub repo for this pipeline. So the history of the SARS-CoV-2, or the nCoV 2019, should change that really, ARTIC Nextflow Pipeline. In February 2020, we had the first cases of SARS-CoV-2 in Wales. And in early March, we were able to sequence and analyze those. And is any microbial genomics presentation complete? If you haven't got a screenshot of the tweet in there. This is Tom, my boss, talking about the first samples that we had at Public Health Wales, where we went from, well, the royal we, I was on holiday, even better. They went from extract to the sequences up on GISAID in about 24 hours, which was amazing. But this was all done, all the bioinformatics here was done by hand, alongside all of the lab work as well. Towards the end of March, it was becoming increasingly evident that we weren't going to be able to sustain that sort of level of sequencing. We had funding from Welsh Assembly Government, and we moved the routine sequencing of SARS-CoV-2 to Illumina, just to increase our throughput and slightly reduce our costs per sample. We do a lot of Illumina sequencing in the pathogen genomics unit anyway, and it made a lot of sense to sort of coalesce on one platform. And then since about April 2020, brief respite in the summer, but we've sequenced more than 500 SARS-CoV-2 every week, and they've all traveled through this pipeline. This is the number of samples we've sequenced each week. So after the great Illumina reagents shortage of early December, we ended up with a sort of 2,000 plus sample week, which was a bit hellish. But everything's now on Illumina, and we're no longer doing the bioinformatics by hand. We've moved everything over to this Nextflow pipeline that I'm going to describe in a minute. And what we wanted, well, the state of play at the time was that the tooling around the Nanopore analysis workflow and the sequencing data through the Nanopore platform was excellent. It was definitely good. So this is Nick and Will's ARTIC package that worked really well for going from sort of sequencer to consensus faster at the end. And it was brilliant because per sample, you would run pretty much two commands, and all the dependencies were packaged in Conda, as Will described earlier. Really great. The Illumina side of the pipeline was a little bit more unpolished. So IVAR, the software existed, but it was quite, as I say, unpolished. It was per sample. You had to run several pretty complicated commands. There were many software dependencies. It was very difficult to come up with an automated approach. So what we wanted to do is build a unifying pipeline in a workflow language called Nextflow, and this is available here. And this would unify the approaches for the Nanopore sequencing and Illumina sequencing. You could run a single command per sequencing run, and it would analyze all of your samples and produce you consensus fastest for all of them. It would bundle software dependencies with the pipeline. It would set some sensible defaults for all of these tools, which have many parameters, and it would be a good idea to set. It was a good idea to set sensible defaults, and also output some human readable files alongside the consensus fasters. So these mostly CSVs that could also be parsed by machine. And these would contain things like QC or mutation or variant calls, etc. So I guess this is my evangelizing bit about Nextflow particularly, or workflow managers in general. So examples of these include, as I say, Nextflow and also SnakeMake. And what they are a programming language that joins the inputs and outputs of bioinformatics. And what they do is they allow you to run the same set of processes on a single sample or on many samples all at once in parallel, using as many computing resources as you can provide it. And why do we want to do that? Particularly in public health bios, we have pretty extensive experience using Nextflow. All of our clinical pipelines for bioinformatics are written in Nextflow. And outside of this, you can write reproducible pipelines because the workflow manager deals with software dependencies and software versions, etc., whether they be packaged in Conda or in containers like the Singularity containers or Docker containers. Workflow managers take care of the scale. So this means you can run the same pipeline code on your set of samples, whether it be on your laptop, on your institutional HPC, or on the cloud. There will be a difference. There will be a time cost associated with running 384 Illumina samples through your laptop, but it will still work. And also on top of that, the workflows of the pipelines are shareable. So we host the NCOV 2019 ARTICnetwork pipeline on GitHub with the intention that it could be used by anyone, either as is or as a jumping off point to build their own pipelines. I promised you horrendous boxes and arrows, and here we are. This is what the pipeline does. So as I said earlier, it coalesces the Nanopore workflow that Will talked about, which is in blue up here. So this is the ARTIC pipeline, essentially the duplex command and the ARTIC minion command, the Nanopolish and Medaka variants of that, and this produces, as already said, consensus sequence, filtered variant calls, VCF, some QC outputs, and it sounds like more are on the way, which is really exciting. And then there is also the Illumina version of this. So both of them take as an input a run directory. So this is from a Nanopore sequencing run. This is from an Illumina sequencing run. They do some read prep. So Guppyplex does some nice stuff with throwing away reads that are too long for the scheme or don't start in the right places. I think trim reads is just a, well, trim galore is just a pretty simple Illumina read trimming tool. Then the next step is variant and consensus generation. So with the Nanopore workflow, this is all handled for you by Will and Nick's excellent tools. Whereas in the Illumina workflow, we map reads to a reference, we trim the primer sequences using IVAR, we call variants, and we make a faster consensus, which is an output here. We generate some QC CSVs, and we also make a variance tab file, which is the same, or the equivalent of Will's VCF. There's also a little bit of extra stuff that we've added on the end. I'll talk a little bit more about this in a minute, which allows you to type samples based on the variants that you find in them. So I'm just going to highlight a couple of key things in this pipeline. I'm not going to talk through it in any great detail because I don't have time, but just pick out a few interesting parts of this. So first of all, preparing references. The pipeline automatically downloads the ARTIC primer schemes repository and grabs the correct primer scheme and reference, though I think recent developments brought this into the ARTIC pipeline itself. So this is due for an update soon. You can also bring your own reference sequence and primer bed file if you're using Illumina, or you can also download it automatically if that's what you want to do. At the end of the Illumina pipeline, we generate a little QC summary CSV. Version you saw Will showing you the multi-QC report, and we make a CSV because you can view it in spreadsheet software if you want, or you can parse it with Python, very easy to do, and it just gives a list of the samples, how many uncalled bases there were in, or the percentage of uncalled bases in the consensus sequence, the percentage of bases that were covered by more than 20 reads, the longest chunk of bases without an N in the consensus sequence, and the number of aligned reads. So you can see here we've got sample 1 and sample 2. They've both passed QC because they've hardly got any N's in them. They've got lots of bases covered, whereas the sample 3 has failed QC. So it's got lots of N's, it's 56% N's. It's only got 43% its covered bases, but it's only got 1/5 the number of reads that align to it. So this makes sense. And as I said earlier, you can generate your consensus sequences, which allow you to share your genomes with the world. QC pass ones only, please. Whether this be via GISAID or GenBank, or you can use them for downstream analyses like lineage assignment. And I think you're going to hear more about pangolin this afternoon. You can do some genetics, and you can do some genomic epidemiology with your FASTA sequences at the end. And then just to finally talk about this variant typing workflow, which we added just before Christmas in response to the proliferation of this variant in the UK, which was is now called VOC 2020-12-01, actually. And this just is an arbitrary name given to a set of mutations, same as what the South African variant was called originally. And to enable you to type a sample, all you need to do is provide a GFF file and a file describing the type. And what you get out is a CSV file of all the samples that belong to each of these types, a summary of the variants that are found in each of the samples and their amino acid translations, and a BCF file with translations too. So what this actually looks like is, this is how you define each of your types. So this is the name of the type, so the VOC. This is how many the proportion of the variants that have to be found in the sample to be able to call it that particular type, and then gene name and the mutations that are associated. What you get out at the end is a little typing CSV that says, okay, for sample 1, we've called it VOC 2012-01 because it's got 15 of the variants that were defined in the file. It's missing 2, but that coverage is over the 0.7 which we use. You also get a nice list of all of the translations of all of the variants, so each gene, the DNA var, and the amino acids translation. So this is just a single slide of how to run the pipeline. I think just shouting Linux commands at you isn't particularly helpful, but all of the information should be in the repo. What I wanted to do with this was just illustrate the value of writing things in a workflow management language. So in order to run this pipeline, all you need is a Linux system. So you could use the ARTIC Lab on SSD for that. The Nextflow executable, which you can download from Nextflow, I think, as far as I know, it will be included on the Lab on SSD, but it's very easy to install yourself. And if you're running on a laptop, you need the Miniconda package manager. And pretty much the prerequisites are the same. Even if you're running on a HPC cluster, you can use Singularity. Many HPC clusters have this installed. You can use Miniconda and then Slurm or another job scheduler. And these are the commands that you'd run to go from, and they run from laptop to HPC. So you'd run the same pipeline on your same sequencer output directory using this command on a laptop. And then all you need to do to run this on a HPC is just indicate that you want to use Slurm, and it will use all of the available queuing system and run all of your samples in parallel, which should enable them to be run much faster. So this is just the Illumina pipeline. If you want to run the Nanopore version, you can use the Medaka flag and just give it the fastQ files, and it will do the same thing. There's very extensive help available if you just use the -- help function. So to summarize, just to round this up, if you're doing your own sequencing, you need a way to go from your sequencing output directory to a consensus genome, because that's the currency of SARS-CoV-2 genomics. Lots of tools already exist for this job, or several tools already exist for this job. The ARTIC pipeline for the Nanopore side and IVAR for the Illumina side, but they can only scale so far. You are running each one manually per sample. There's only so much typing one individual can do if you're sequencing 2,000 samples a week. So our response to that was to write a Nextflow pipeline, which allows you to run a single command to turn a whole SARS-CoV-2 sequencing run into consensus genomes, and also provide you with some QC and some other sugar if you're interested in that. Then as a sort of general point, workflow managers can handle the software dependencies so that your analysis is reproducible. Anybody can run the pipeline and end up with the same versions of all of the dependencies, so that if you run your pipeline on HPC, you'll run it on a laptop, or somebody else wants to run the pipeline. The workflow manager means that this is scalable, portable. You can run it on 10 samples or 10,000 samples. You just pick the hardware, laptop, HPC, cloud. And finally, just to say that the pipeline is available here. If you have any problems, if you have any issues, if you have any features to request, please just open an issue, and hopefully when Illumina run out of sequencing reagents, I'll get some time to make some changes and add some features if that's something that you are interested in. And with that, I'll finish there. Thanks very much.
Thank you, Matt. Let's give you a round of applause in the usual way. So we've got a couple of questions in the chat for you. Several questions coming in quickly. Firstly, a question about low frequency variants. So will IVAR, I think this relates to the Illumina pipeline particularly, will IVAR call low frequency variants within a sample, and will it also give confidence scores?
Yes and yes. All of this is very tunable within IVAR. So IVAR will allow you to set a minimum coverage to call a variant. If your coverage drops below that, it would just be called an N. And it would also allow you to set a threshold over which, an alt fraction threshold over which your the base will be called the alt. If your fraction of alt bases are underneath that threshold, IVAR will call your consensus as a mixed base. So it keeps everything in as much as it possibly can, as far as I'm concerned.
Okay. this wasn't a question, but can you just comment on intra-host variants with the Nanopore pipeline as well?
No.
Okay.
I'm afraid I don't know enough about it to make an informed comment.
Okay, that's fine. All right. So next question. Let's take this one from Federico. It's actually probably a question for Matt and for Will. So which minimal depth do you normally use for making a base call or put an N in the consensus with IVAR for the Illumina pipeline? And then maybe Will can answer for the Nanopore pipeline as well.
So as far as I'm concerned, the IVAR or the Nextflow IVAR pipeline will only call an N with testing me now. It's either 10 or 20x coverage. So this is it's written in the help, the default, but I haven't looked at that today.
I think it's 10.
Yeah.
Certainly for COG, 10 for Illumina, 20 for Nanopore is the default.
Yeah, I knew I got 10 and 20 from somewhere.
Yeah. Okay, so I think that's answered that question. And Nabil's also confirmed that in the chat. A question from Sergio. For updating FastAid consensus in GISAID, which is the best option, the IVAR raw consensus or a post-processing consensus?
I don't really understand. What's a post-processing consensus?
Maybe Sergio could unmute and ask his question directly if he wishes to. Let's see. Sergio, do you want to? If not,
feel free to elaborate more
or just write it in the chat if you prefer. Okay, we'll come back to that one. A question, oh, here we go. Okay, so the question is, what would you recommend if we want to look for co-infection with other potential pathogens, i.e. bacterial pathogens in COVID?
Difficult question, really. I think the pipeline is focused very much on SARS-CoV-2. It deals with amplicon data that is targeted specifically towards searching for SARS-CoV-2. It might be that there is some good mileage in some metagenomic sequencing for other pathogens at the same time, but that's not something that the Nextflow pipeline would handle. There are plenty of excellent other pipelines written in workflow managers for doing metagenomics or that sort of thing. A good place to start would be the Nextflow core team, who have lots of pipelines available for doing various things with genomics and metagenomics.
It's worth emphasizing, isn't it, that the multiplex PCR method will only ever detect the pathogen of interest. And so if you're using a SARS-CoV-2 amplicon scheme, it's only going to detect SARS-CoV-2. Josh did mention the idea that you can put in multiple primer schemes to find multiple pathogens, but you would effectively run the pipeline separately for each pathogen of interest. As you say, otherwise, if you're looking for co-infections, you're really thinking about a metagenomics type strategy, an untargeted strategy, which is not really the subject of these pipelines and pushes you into the metagenomics space for software, things like Kraken and Centrifuge and all those kind of things. Nabil's helpfully put up a link for some metagenomics options for those who are interested in doing that. Another question, Ricardo asks, does the pipeline, so can it run systematic processing in large data sets? And if so, what is the average running speed?
Yes, it's a good question. This is sort of contingent on the amount of resources that you can give to your pipeline. I can give a few illustrative figures from my experience with our cluster at PHW, and I think we have a relatively small Slurm cluster that lives in the lab. I'm sure the lab people are really pleased about. and it's some 150 cores or something. And we can process a whole 384 NextSeq run of SARS-CoV-2 in about 40 minutes. And this goes all the way from the sequencer finishing to the typing, the consensus fasters, everything else. Obviously, if you're running this on a laptop, it's going to take much longer because you don't have the amount of resources to be able to schedule multiple things in parallel. But yeah, if speed is if speed's really important, then you can always run it on the cloud or whatever by changing a few bits of config with the workflow manager, and you can make it as parallelizable as you'd like.
Excellent. And there's a bit of clarity on that question from Sergio. So I think he's saying in his lab, he uses the IVAR raw consensus and then processes it with stricter filters. But he would like to know if the output of the IVAR consensus is good enough on its own to send to GISAID.
To be honest, I don't see any problem with that, as long as you're not hassling the poor curators at GISAID with sequences that are 90% N. Then I think sending sequences with mixed bases in or RUPAC codes in is perfectly fine. At least it retains all of the information about what variants might be in that particular sample. But agree with Nabil. This is a matter for your own local QC, and I hope it sounds like his talk is going to cover some useful stuff about how to QC your own sequences.
Absolutely. All right. Well, thank you very much, Matt, for your great talk and also for your work on that pipeline that I know has been extremely useful for many groups in COG and elsewhere.